Hello and welcome to Biomaterials in Action, a special programme that investigates the problems of living with and managing debilitating and painful chronic dermal wounds. In particular, we'll be examining the work of the DERMA project, which is comprised of a group of academic researchers and clinicians co-funded by the European Union Interreg II CESE programme. DERMA was formed with the specific goal to improve the welfare of patients suffering from these distressing wounds. The project recently announced the results of its 4-year work programme to develop new materials intended for management and treatment of these chronic dermal wounds, including ulcers of the leg and foot. In context, it's estimated up to 2% of the EU population suffers from these painful and unpleasant wounds at any given time, with this figure increasing greatly with age. The management of these by healthcare workers costs the EU €6.5 billion each year. Chronic ulcers can also take a long time to heal, require extensive management by healthcare workers, and despite this, may still be subject to poor healing outcomes. Of special concern in the management of these wounds is the intermittent eruption of painful and damaging bacterial infections within the wound site, and the unpleasant odour they release. Indeed, some patients suffering from such malodorous chronic wounds may experience embarrassment, depression, and social isolation. I asked Professor Simon Toe from the Portsmouth NHS Trust to describe his experience of caring for patients with malodorous wounds.
Hi, I'm Simon Toe, consultant surgeon at Portsmouth Hospital University NHS Trust. I manage surgical wounds every day. Smelly wounds or infection or necrosis is a common problem for our patients and their care teams, both in hospital and at home. They are a source of deep embarrassment for our patients and can lead to patients isolating themselves, leading to mental problems like depression. On wards, the smell of wounds is a major problem for our nurses and other patients and has been shown to reduce appetite and increase anxiety. Humans have an inherent aversion to bad smells, and so these patients tend to get less care and attention. It therefore follows that removing the odor can be transformative, ideally by treating the infection or removing necrotic tissue. But where this is not possible or there are delays between dressings, we need a better way to manage the odour.
Thank you, Simon. To give further insight into the experiences of those living with chronic wounds, here is a short scene depicting a typical conversation between a patient with a foot ulcer and a podiatrist treating them.
I hope you don't mind me asking, but you're not your usual self today. Are you okay?
To be honest, not really. Ever since I had this wound, I keep thinking it's going to be like the last wound I had. That took 6 months to heal. It was a dreadful experience. Just everything seems to have gone wrong. I can't do the things that I used to do. I don't go out anymore. I don't go out on Fridays anymore. I just feel really cut off.
You mentioned that you were worried about the odour from your foot.
Yes, I think it smells. I hasn't been near a bath or a shower for ages. I'm really worried that it smells.
We can see then that these wounds can cause great concern for patients, to the extent that their confidence and quality of life can be significantly adversely affected. Fortunately, some products are available to help alleviate the malodor of chronic wounds, and these can work well for easy-to-access wounds. However, certain types of wounds can be very difficult for clinicians to dress with conventional materials. I asked experienced podiatrist Rachel Force from the Leaf Hospital in Eastbourne on the south coast of England to explain.
Patients want to get on with their quality of life, really. They don't want to be restricted, they don't want anything that's going to cause them discomfort or pain, they want to keep active, they require something that is pain-free to remove and apply. It's comfortable, it conforms to the area where you apply it, where the wound is. not too bulky. They want to be able to carry on with their day-to-day life, and it's not going to be too restrictive. So they don't want it to be too obvious, and they just want to carry on with day-to-day life as much as possible.
Bacterial infections of chronic wounds are a significant clinical problem. I spoke with Professor Stan Munstre, a plastic surgeon with expertise in treating burn injuries and chronic wounds, from the Department of Plastic and Reconstructive Surgery, Ghent University Hospital, Belgium, to explain his approach to the treatment of skin infections.
Burn wounds belong to the group of the chronic wounds, and burn wounds, in my opinion, are more at risk of becoming infected. For this reason, antiseptic and antimicrobial agents are probably more important than in any other wound. I think burns do have two specific characteristics. One, first of all, is that usually the skin is damaged over pretty larger areas, which means that these wounds are more than other wounds at risk of becoming infected. And the second thing is that sometimes the healing takes a long time, and therefore also the risk of infection at a later stage is higher probably than other wounds. This has, by the way, been shown by the fact that long-standing burn wounds or burn wounds that take a long time to heal are always full of biofilm, which makes it even more difficult for the antiseptic or antimicrobial agents to become effective. But the conclusion is that almost all wound dressings for burn wounds do have an antiseptic or antimicrobial agent included, usually already by rinsing, but also involved in the wound dressings itself. Since the wound dressings are often very painful and they cause a lot of discomfort and require a lot of work to be changed on a daily basis, it has become more and more important to have antiseptic agents with an extended or a slow release, which means that the antiseptics still remain effective, but you don't have to change the wound on a daily basis. Of course, also the degree of moisture of the wound is a very important aspect for the treating of these burn wounds.
Thank you, Stan. It's apparent then that chronic dermal ulcers are a common and serious problem for society. They are painful and can produce unpleasant odours that are distressing for the patient. Intermittent and difficult to treat infections can erupt within these wounds, causing the condition of patients to deteriorate, even to the extent that an infected limb could be lost. It is in this context that DERMA Consortium challenged itself to develop new materials for the improved management of such dermal wounds. After the break, the DERMA team will describe in detail the development of these materials.
DERMA is funded by the European Regional Development Fund under the Interreg 2Cs program. 1 to 2% of the population will experience a chronic non-healing wound in their lifetime. The objective of DERMA is to develop new materials for the management of chronic non-healing wounds.
Before the break, we heard that chronic dermal ulcers are a major problem for society. They are painful, produce distressing malodours, and they can worsen due to difficult to treat intermittent bacterial infections. We also learned that a consortium of EU scientists and designers working on a project called DERMA had set themselves a challenge to develop new materials for the management of these problematic wounds. I asked the DERMA project leader, Dr. Ian Allen, of the Centre for Regenerative Medicine and Devices at the University of Brighton, to describe the project in more detail.
Well, the DERMA project built on previous collaborations between researchers from the Universities of Brighton and Portsmouth in the UK and the University of Ghent in Belgium. These projects being concerned with the development of biomaterials for dermatological applications. For DERMA, we also recruited Eurosante from Lille in France, which is an agency that facilitates the tech transfer of healthcare products to market. The DERMA consortium was multidisciplinary in nature. It included polymer chemists, microbiologists, material and digital designers, tech transfer specialists and clinicians. And we realised as a consortium that in terms of collective expertise, we were in a good position to develop materials that could potentially improve on what was already available in the field of wound care, especially in the management of chronic dermal ulcers. So the consortium decided to intervene in this field in 3 areas by making materials intended to 1. Help manage the unpleasant odours produced from dermal ulcers, 2. Tackle bacterial infections within these wounds with improved efficiency, and 3. Produce a material designed to give a visual indication that a bacterial infection is taking hold within a wound.
As Ian mentioned, the DERMA consortium has been developing new materials that can manage the unpleasant odours associated with chronic wounds, whilst possessing favourable physical properties applicable to wound care management. I asked Ian about the materials he's been developing at the University of Brighton for wound odour control.
The DERMA team was working on a number of materials to help manage malodour in wounds. One of the most promising materials developed at the University of Brighton was composed of agarose being derived from seaweed. Many people will be familiar with agarose gels, sometimes referred to as agar, and these being used for a variety of purposes, especially in scientific laboratories and for culinary uses. What is less well known about these agarose gels is that they can be dried to form transparent and very brittle films. But to overcome this brittleness, a non-toxic plasticizer can be added to the gel mixture before drying, producing films that are very much like cling film in appearance. So these agarose films are non-toxic and they are biodegradable. In the context of medical materials such as wound dressings, they have low adherence, so they won't stick to the skin or indeed the cells within a wound. The films are breathable, that is, they allow the passage of water vapour, meaning that if films are held close to the skin, excessive sweat and moisture won't build up, which can potentially lead to tissue destruction, known as maceration. For wound odour capture, we incorporated activated carbon particulates into these plasticized agarose films. Activated carbon has a well-known capacity to capture or absorb small molecules on account of its very high surface area to volume ratio. So once developed, these activated carbon containing plasticized agarose films were assessed for their ability to capture malodours typically found in chronic wounds. We also incorporated a special polymer into some versions of these films, one which has a remarkable ability to absorb water. This gave the option of having a film that is able to swell greatly in the presence of moisture, with the intended function of absorbing excessive exudate from a chronic wound, in addition to the odour capturing properties. So the more of the absorbent polymer that is present in the initial formulation of the material, the more moisture can be absorbed. So in theory, we could tailor these materials to wounds with differing exudate volumes, from relatively dry wounds to runny or sloughy wounds. We're very pleased with the feel of these materials. They're easy to handle and they can be cut to shape with scissors. The films are heat sealable, so this allows the activated carbon to be fully enclosed in kind of a sandwich arrangement, with laminates of our plasticized agarose enclosing an inner activated carbon agarose layer to prevent direct contact of the activated carbon with the wound bed.
Thank you, Ian. As we have learned, some of the unpleasant odours from chronic dermal ulcers are caused by the action of bacteria in the infected wound. Therefore, the DERMA Consortium also set itself the task of trying to improve the treatment of such infections by developing materials that can release, over an extended period of time, antimicrobial agents that can inhibit or kill the bacteria responsible for these painful infections. The materials developed to deliver these antimicrobial agents necessary to tackle these infections have been produced by a method known as electrospinning. I asked Professor Peter Dubruel of the Polymer Chemistry and Biomaterials Research Group at the University of Ghent, Belgium, to describe this process in more detail.
So within the DERMA project, we have had the pleasure of applying an innovative technique, which is called electrospinning, with the aim to produce polymer mats. The reason for doing so is related to the fact that in our bodies, cells are embedded in the so-called extracellular matrix, and this matrix is also composed of these randomly distributed fibers, which are then composed of biopolymers. During this technique, an electrical field is applied onto a polymer solution, and via a complex cascade of events that leads to these electrospun mats. The beauty of the technology is that both small and large scale materials can be developed at a high speed, and more importantly, they can also be loaded with biologically active compounds, which actually then enable compounds like antimicrobial compounds, which can then be released over time when they come into contact with the wound bed. So the versatility can be found both in the type of polymers that are applied, but also in the antimicrobial agent, and of course through the technique of electrospinning, which tries to mimic the so-called extracellular matrix to the highest extent possible, as this technique enables us to produce materials with a high, very high even, surface to volume ratio.
Thank you, Peter. In the following footage, Dr. Arne Mignon, also of the Polymer Chemistry and Biomaterials Research Group at the University of Ghent, Belgium, demonstrates the electrospinning process in the laboratory.
To be able to electrospin something, you first have to make it. So polymerization, to create the materials which we will electrospin in the next step, are actually made with these systems. At the beginning we have to make our materials, then we make some sheets out of it, we test some with mechanical properties, we do some other tests for characterization, and afterwards we will mix them in a solvent, we put them in the electrospinning, we electrospin the fibers. Then with electron microscope, we look at them on a submicron scale. And then we go to the next step, where we can incorporate the odor absorbing or the color indication.
We have seen how the electrospun materials are produced in the laboratory and that they are extremely thin. I asked Professor Sandra van Vlierberg, also of the Ghent Group, to explain the advantages these fibres possess.
So we can control wound exudate uptake with our hydrogels by using different chemical tools. One aspect which we can use is the length of our polymer chains. If we use polymers with a higher molar mass, which implies a longer chain length, the material will swell more, and this means that it can also take up higher amounts of wound exudate. If we use polymers with the shorter chain length, the material will swell less and will take up lower amounts of wound exudate. A second building block or a second methodology we can exploit is the degree of network density. If we use higher network densities and a higher cross-linking degree, our network will swell less, and as a result, it will be more appropriate to serve lower exudating wounds or less exudating wounds. If we have a network which is less tightly cross-linked, then it means it can swell more and is also more appropriate to apply onto wounds which show a lot of wound exudate. This wound exudate is also crucial because it contains a lot of growth factors which can aid the wound healing process. And therefore, it's important to also keep this exudate close to the wound throughout the healing process. And that's why your hydrogels really need to be tuned towards the application. And with this tool set of chemical building blocks we can exploit, it means we can create hydrogel dressings which are appropriate either to treat less exudating wounds or more exudating wounds in line with the wound healing profile required for the application.
Thank you, Sandra. As well as trying to tackle existing wound infections by developing materials that can release antimicrobial agents, the DERMA team also wanted to produce a material that would give a visual early warning that an infection was in the process of developing within a wound, enabling much earlier intervention by healthcare workers before a serious infection can take hold. I asked Dr. Allen to describe the work undertaken by the DERMA team to produce such a diagnostic material.
Well, we wanted to produce a material that would give an easy to understand signal that an infection is starting to develop within a wound, indicating that intervention is required to prevent the condition of the wound from deteriorating. Early in the project, when deciding what our material should be designed to detect, we considered a number of factors that have been reported to be indicative of bacterial infection within a wound. These included changes in wound pH, temperature, and also the presence of specific molecules within a wound. In the end, we decided to detect bacterial activity directly using a special dye that changes colour from dark blue to bright pink in response to the presence of growing bacteria. So this dye, which is called rosazurin, has been used for many years to detect bacterial growth, and it was first used to monitor the spoilage of milk. The teams from the universities of Brighton and Ghent were working on means to incorporate this dye within their candidate dressing materials. At Brighton within the agarose films previously mentioned, and at Ghent using their electrospun fibres. It was important that when the dye was added to these materials that it would be retained efficiently within them and not leach out. In other words, we didn't want the dye to fade from the materials and to leach into the wound bed. It was also important that the expected blue to pink colour change could still occur when the dye was incorporated into these materials in the same way that it would do when it was in its free form. This proved to be quite a challenge. After all, there doesn't appear to be a similar dressing on the market. However, ultimately, we think we've made good progress.
It's obvious that any material intended to be applied to wounds must be acceptable for the patient. Similarly, healthcare workers would need to be convinced of the clinical need for the material and be able to apply it with relative ease. I asked Dr. Cressida Boya of the Centre for Creative and Cultural Industries at the University of Portsmouth how the DERMA team was able to understand the needs of patients and clinicians.
Throughout the project, we've hosted focus group discussions with patients, carers and clinicians to talk about wound dressing requirements. The University of Portsmouth Ageing Network identified and invited patient representatives to help us understand the problems associated with chronic wounds from the patient perspective. This helped us to identify unmet needs and research objectives for new dressing designs. Fashion and textiles expert Tom Cluley visited the dressing clinic at Leaf Hospital, and consequently Tom and Professor Joan Farrer were able to advise DERMA scientists on desirable material properties for difficult to dress areas of the body. Scientists in the Faculty of Technology went on to test and assess the DERMA materials according to these parameters. We produced a short empathy film, both as a touchstone for the research team and also for public dissemination. This film was scripted following a visit to the dressing clinic. As well as being helpful for DERMA researchers, there are plans to use the film as a teaching tool for carers and clinicians to help them understand and empathise with patients. Health psychologist Dr. Ms. Al-Abadi is carrying out research into the lived experience of people with chronic wounds and the acceptability of new dressing materials.
Thank you, Cressida. It's evident that there were several technologies with various intended functions being developed in parallel by the DERMA project team, involving collaborations with several laboratories. I also asked Cressida about the involvement of designers and digital animators in the project and how they were able to help the researchers to stay focused on the scientific goals of the project.
As you can probably tell, one of the things we're very interested in at Portsmouth is how we can utilise creative methodologies for health and wellbeing projects. Creative outputs can be used as research tools and teaching aids, and for disseminating complex research findings to non-expert audiences and in an easy to understand and engaging way.
One member of the DERMA team we have not heard from as yet is Eurosante, based in Lille in France. Eurosante is an agency dedicated to tech transfer and business development in the life sciences sector. I asked Xavier Martin Benloche about the role Eurosante played in helping put the DERMA consortium in touch with people and businesses with an interest in wound dressing development.
Yes, thank you. Eurosante is a leading agency here in the north of France that's dedicated to bringing new healthcare innovations to market. In terms of the DERMA consortium, we've been involved since the beginning to bring this market-centered design to the project and so that all innovation and all products developed throughout the process have this market approach from the beginning, so that we can then easily translate all discoveries and innovations to our industrial partners, but also and eventually to the patients. One of the things we did was participate in two leading partnering events in the healthcare sector, which are BioFit and MedFit, which are organized by Eurosante, and which bring together researchers, clinicians, technology transfer offices, industrials, and startups together in order to foster innovation in such a way that new collaborations arise and that these new innovations can get to the market and to the patients.
Thank you, Xavier. We've now seen how the DERMA consortium has been engaged with the development of novel technologies for the improved management of chronic dermal ulcers. In the final part of the programme, we will discover the key results of the project and discuss their significance towards better outcomes for patients suffering with chronic dermal wounds.
DERMA, Design of Enabling Regenerative Materials for Aging. DERMA is an EU Interreg 2Cs funded project running from 2016 to 2020. Partners are the University of Brighton, the University of Portsmouth, the University of Ghent, and Eurosante. DERMA engages the development of new healthcare technologies to improve the quality of life of the ageing population in the 2Cs area. The objective is to develop new interventions for the management and treatment of dermal ulcers and related skin conditions. Dermal ulcers are a source of physical pain and discomfort, can lead to further medical complications, and can liberate offensive odours that are distressing to the patient and may lead to social isolation and depression.
I think it's an exciting thing to be involved in. I think I'm part of a small part looking at the project, that is, looking at the antimicrobial aspects, but talking to people outside of my field is very exciting to understand that all this work's going on. And if you just come together in meetings like this, then you can really come up with something quite special, I think.
To speak to people that actually use wound dressings, to speak to patient representatives, to speak to people that are involved in the regulatory side of things or have experience of that is incredibly useful. And those people don't often all get in a room together. So from my experience, from my perspective, it's been a great experience.
Listening to some of the speakers today, it's solving the issues that are currently there, making things, that wounds can take a long time to heal, and being able to speed things up through technology and through design, it's got to be a benefit for everybody.
DERMA will utilise the experts of clinicians, scientists, designers, creative technologists, healthcare providers and business entrepreneurs and support services to develop new technologies to address patient needs, reduce healthcare costs and improve patient outcomes. On Tuesday 26th of September 2017, the DERMA team made the journey to Ghent for stakeholder and industrial matchmaking events. The events hosted approximately 130 attendees and were held as part of the Advanced Materials for Biomedical Applications Conference 2017. This conference was to celebrate 10 years of polymeric biomaterial research in the Polymer Chemistry and Biomaterials Research Group at Ghent University. The organisers of the event, the Polymer Chemistry and Biomaterials Group, are partners on DERMA. Principal investigator Dr. Ian Allen from the University of Brighton was invited to give a presentation about novel materials for the management of chronic dermal wounds. Dr. Cressida Bauer from the University of Portsmouth presented a poster entitled DERMA, Wound Care by Design, Computer Animations for Public Engagement. And Dr. Arne Mignon presented a poster entitled Novel PEG-based Hydrogel Building Blocks for Wound Healing. Also at the event, Eurosante, DERMA project partners, hosted an industrial matchmaking event. This was followed by a DERMA boat trip to see the sights of Ghent. On the 13th of September 2018, DERMA hosted a series of presentations at the European Society for Biomaterials conference in Maastricht. The theme of the conference was Materials for Life, which expresses the challenge the field of biomaterials is currently facing to provide effective and affordable biomaterials-based methods to repair and regenerate damaged and diseased tissues and organs. The presentations included a variety of experts in the field of wound dressings and management. The European Society for Biomaterials is a non-profit organisation at the forefront of the scientific community, determined to tackle unmet clinical needs by means of advanced materials for medical devices and regenerative medicine. On the 4th to the 5th of December 2018, DERMA hosted a stand at BioFit in Lille. BioFit is the place where academia and industry collaborations get started. It is the meeting point in Europe for tech transfer and for sourcing early-stage innovations stemming from public research institutions, academic spin-offs, and emerging biotech companies. Together with big pharma, biotech and diagnostic companies, BioFit operates as a platform to build partnerships for all public and private sectors. BioFit is also the European marketplace for pre-seed, seed, and Series A investment in life sciences. Eurosante, a partner in the DERMA project, runs BioFit and is involved in hosting the conference. The University of Portsmouth attended to make contact with people useful for the next stage, talking to IP and cytotoxicity testing company to help do early stage trials on the product.
Welcome back. Previously, we heard about the materials that the DERMA team had been developing to help manage chronic wounds, including a material to prevent unpleasant odours emanating from a wound, a material loaded with an antimicrobial agent to gradually permeate a wound and control infection, and a material intended to change colour when a new infection develops within a chronic dermal wound. It is now time to see what the DERMA team have achieved. Firstly, I asked DERMA team researcher and podiatrist Rachel Force of the Leaf Hospital to describe the results of a study assessing performance of a DERMA odour-absorbing material produced at the University of Brighton.
So wounds associated with inflammation and bacterial contamination can liberate malodorous gases from the wound bed. These are substances called volatile organic compounds. These compounds come in different structures and can include thiols, ketones, alkenes, dimethyl polysulfides, and in association with Pseudomonas aeruginosa, a specific volatile organic compound called 2-aminoacetophenone. Early in the DERMA project, a study investigating the ability of an activated carbon-containing agarose film to absorb malodorous thiols was conducted. It was found that this DERMA material in the proximity of thiols was able to significantly reduce their presence in both liquid and in air. The team then decided to investigate the ability of the DERMA material to absorb 2-aminoacetophenone. This is one of the malodorous volatile organic compounds produced by Pseudomonas aeruginosa in an infected wound, and it is associated with having a significant impact on patients' health and well-being. After obtaining ethical approval, 53 volunteers were asked to rate the odour from 6 randomised pots, in which were placed samples that either contained no odour or the odour from 2-aminoacetophenone. The samples were either covered with the DERMA activated carbon containing material, a control dressing without the activated carbon, or no covering at all. The participants were unable to see the contents of each pot, and they were then asked to sniff each container and to state if they could detect a strong odour, a slight odour, or no odour at all. The participants were able to sample each container up to 3 times during the study to enable them to quantify their response. Once decided, the participants' response was recorded by one of the research team. The results indicated that the DERMA activated carbon material was able to significantly reduce the odour present in the laboratory testing procedure as assessed by healthy human volunteers. This is indicated by the significant reduction in the bar height indicated here in the very last column on the chart. It can also be seen that the score is as low as that of the sample with no odour, also covered by the DERMA activated carbon material. This indicates that the activated carbon material was able to eliminate any background odours, such as the plastic used to house the samples, when compared with the samples that had no covering or the control material.
Thank you, Rachel. The following animation gives a representation of how this DERMA material may be applied to a patient in future to help manage a malodorous wound.
A patient has presented with a chronic dermal ulcer. A dressing is required to cover the wound effectively and comfortably in order to help it heal. The DERMA material is flexible, breathable, conforms to the contours of the skin and can be cut to the desired shape, potentially making it suitable for application between the toes, round the heels and other hard-to-dress areas. Chronic ulcers can liberate offensive odours. The DERMA prototype contains activated carbon particulates to remove unpleasant odours produced in the wound. By preventing wound odours from leaving the dressing, it is expected that patients' stress and anxiety will be greatly reduced.
We learned earlier about electrospun fibres and how the University of Ghent has been developing these for the controlled release of antimicrobial agents to help control infection in a chronic wound. I asked Dr. Tom Gaysons of the Polymer Chemistry and Biomaterials Group at the University of Ghent to describe the results of this work.
One of the compounds that we started integrating in our electrospun dressings is povidone iodine, and povidone iodine could then be released slowly from these electrospun mats. In contrast with the current commercially available wound dressings on the market, our material was designed to be slow release. So most materials that are on the market today, the wound dressings that are on the market today, they actually have a burst release. So what does that mean? It means that in a very short time frame, all the antibacterial is released in like half an hour, an hour.
So in this graph we can actually see the comparison between DERMA dressing and the commercial dressing in terms of released iodine in light blue and the inhibition zone in dark blue. So here we see that the DERMA material releases less iodine than the commercial dressing, where we see a very big burst release for the commercial dressing. But the inhibition zone of the commercial dressing is a lot smaller than for the DERMA dressing. So this means that the DERMA dressing will have a more prolonged effect on the wound in light of antibacterial properties. In this figure we can see the comparison between the DERMA dressing and the commercial dressing in terms of half-life. So what is half-life? Half-life means the time that is needed to get half of the iodine concentration leached out. So we can see that for a commercial dressing it only takes 10 minutes before half of the iodine is released into the environment, whereas for our dressing it takes about 2.5 hours to get to half of the concentration of iodine, which means it's a slower release of your iodine in the wound.
One of the big goals of this project was to actually make a wound dressing that was able to release iodine in a longer time frame, so from 24 hours to 48 hours or even longer if possible. So with our material we obtained this result, so we could actually release povidone iodine over a time frame of 24 hours to 48 hours, making it ideal and for a wound dressing, because then it means that doctors and nurses have to reapply the wound dressing much less frequent than the available dressings on the market today. The material itself, so the AUP, the hydrogel material, is extremely hydrophilic, and because it's very hydrophilic it's binding a lot of water, and therefore it can actually take up 3 to 6 times its own weight in water, making it an ideal material to use for wound dressings. In contrast to current commercial available materials, it can actually take 3 times more water up than the commercially available ones.
Thank you, Tom. Here we can see a computer animation that depicts how such a material could be applied to a wound in the form of a dressing.
A patient has presented with an infected chronic dermal ulcer. A dressing is required to cover the wound effectively and comfortably, and to deliver antimicrobials to tackle the infection in order to help it heal. The DERMA antimicrobial material is flexible, conforms the contours of the skin and can be cut to the desired shape, potentially making it suitable for application between the toes, round the heels and other hard-to-dress areas. Bacterial infections within an ulcer require to be managed to allow healing to occur. The DERMA prototype contains an antimicrobial agent to tackle infection. The novel DERMA material can release the antimicrobial for a prolonged period, thereby controlling the infection with greater efficiency and potentially reducing the need for healthcare workers to change the dressing as often as is currently practiced.
We also learned earlier about the DERMA team's work in developing a material that would change colour in the presence of an emerging bacterial infection. I asked Dr. Ian Allen to describe the results of this work.
The DERMA team was working on two versions of a diagnostic dressing using very similar materials. One, a film form based again on the use of plasticized agarose, and an electrospun fibrous version. But I will describe here the results of the agarose film form. So the films were tested against well-known problematic bacteria responsible for chronic wound infections, including Staph aureus, E. coli and Pseudomonas aeruginosa. And the presence of bacteria was indeed able to be detected by these films by way of a stark colour change from blue to fluorescent pink, caused by activation of the dye held within the film. We found that we could immobilise the dye in sheets of a non-toxic polymer matrix and encapsulate these sheets within laminates of our swellable agarose films. This allowed our diagnostic material to take up fluid that contained bacteria and then respond by producing a colour change. We anticipate that this would also occur if our material, subject to ethical and regulatory approval, is applied to infected wounds on patients. We made a technological breakthrough quite late in the project, and though we haven't yet proven this, we hope that in future it will be possible to calibrate the material to change colour in a predictable way according to the thickness of the dye-loaded matrix and also the concentration of bacteria it is exposed to. This could allow for a range of diagnostic materials that would be able to indicate different severities of wound infection. At present, depending on the formulation used, our films start to change colour after a few minutes of contact with growing bacteria, and then the reaction progresses over the next few hours until the colour transition is complete. In this time-lapse video, the diagnostic material on the right has been placed on a lawn of E. coli bacteria growing at body temperature. The material on the left is not in contact with bacteria. The colour transition from blue to pink is shown developing over a period of 2 hours, 20 minutes. So the next phase of the work will be to fine-tune and calibrate this response.
Thank you, Ian. The following animation depicts the colour change that an ulcerated patient will notice if an infection erupts within their wound.
A patient has presented with a wound of uncertain prognosis. A dressing is required to cover the wound effectively and comfortably in order to help it heal. Bacteria within the wound are starting to multiply. The dividing bacteria produce chemical changes within the wound. The DERMA material is a transparent film infused with a special dye which reacts to the increasing numbers of bacteria. The molecules of the dye respond by changing colour, indicating that a bacterial infection is developing within the wound. The colour change will alert healthcare workers that further intervention is required to manage the wound.
We have seen that the DERMA team has produced materials that appear to hold much promise for future treatment of patients with chronic wounds. But what hurdles are still to be overcome before these materials are available in clinic for the treatment of patients? I asked Professor Matteo Santin of the Centre for Regenerative Medicine and Devices at the University of Brighton about the regulatory steps a potential dressing material must satisfy before being approved for patient use.
The regulation of medical devices is obviously looking at both the safety of the patient as well as the industrial feasibility. In the case of wound dressing, these are devices that have been historically categorized as Class 2B medical implants. The evolution of the regulatory framework, however, has made their approval to the market more stringent. So from looking at the toxicity aspects of the materials that have been considered by the manufacturer, by the R&D project, to their validation in terms of in vivo testing, clinical trials, and finally the efficacy. It is obviously a long and rather expensive process requiring investment. But basically, the need for medical devices, particularly wound dressing of a new generation, is becoming more and more, has been becoming more and more relevant if considered that the clinical need is widening in the aging population, as well as with the higher incidence of some diseases like diabetes, leading to foot ulcers and problem with the chronic wounds. The DERMA project fits very well in this process of development, because alongside the innovative approach can indeed is already basically developed and designed to fulfill the more stringent regulations that I've just mentioned.
Thank you, Matteo. Satisfying the regulatory demands on a new material before bringing it into clinical use does seem to be a formidable challenge. Therefore, I asked Dr. Brian Griffiths of BG Healthcare about his experiences of doing just that, developing a wound dressing that did make it to clinic.
I was very fortunate in that I've been involved in teams that have developed multiple dressings that have made it to market. And that process can be quite a long-winded process. It involves lots of different facets of development, not just the development of the dressing itself, but aspects around regulatory, aspects about clinical evidence. There are lots of elements of manufacture that need to be resolved when you're in development, and a whole host of other things around stability, etc., and packaging. And all of those things need to be considered before you get to the point where your sales and marketing team are happy then to kind of take that product and get it into a position for launch. So there are lots of different aspects involved in the development of a dressing. I think lots of people consider that the development is a very technical thing, and certainly the inventors get very close to the product, and that's all they see very often. If you're fortunate enough to be in a large organization, there are other people worrying about the regulatory and clinical aspects of the dressing, whilst you as an inventor get to stick with the product, make many iterations of your design, constantly improving that design and constantly testing that design before you get to a point where you are then ready to get that product into a clinical setting. So the development phase is long and complex, and very often in large organizations involve multiple teams to make that final project a reality, or make that final product a reality.
I also asked Brian what advice he would give to scientists who are developing materials that they believe have great potential for patients.
So my advice really to any new designers is to be bold. We are now in a phase where advanced wound care products really exceed those of traditional products. And so designers have been kind of pushing this space for a number of years now. My advice, I guess, would be to think about things like smart bandaging that really tells you something about the wound interface. I think that is a very exciting kind of area to be working in, and kind of gives us another kind of option when we think about treating chronic wounds. So that's the advice: just be bold in your thinking and push the boundaries if you're able to.
Thank you, Brian. Finally, I asked project leader Dr. Ian Allen to sum up his thoughts on the success of the DERMA project and the benefits he hopes it will bring to society.
DERMA was certainly an ambitious project, and I'm really pleased with what we managed to achieve. Some of the technologies will continue to be developed, perhaps with the help of additional investment with a view to getting these to market. Only by doing this can the expected benefits for patients be realised, and of course that is why we undertook the work to help improve the health and well-being of patients with chronic wounds. I'd really like to thank the Interreg 2Cs programme for providing so much support and encouragement to the DERMA team during the project. It's been a great experience to work with so many talented colleagues working across disciplines and from different countries, all on a common objective. Personally, I really hope that the DERMA consortium continues to work together despite the very different political landscape that exists at the end of the project compared with the start. I think people will always find a way to work together if the will is there, and of course with work comes friendship too. I'm quite sure that across Europe there'll be other groups of workers who share similar feelings about their own projects, and that we'll see a determination to maintain the links and friendships that have been so valued by us all.
Thank you, Ian, and thank you for watching. Goodbye.
This is where DERMA comes in. This activated charcoal product can be incorporated into dressings or wound management systems like negative pressure dressings. Can you imagine what a difference it will make for our patients? It'll enhance their quality of life immeasurably, and I look forward to DERMA in clinical use one day.
I can't detect any odour, but if it helps your peace of mind, we could use a dressing that's designed to remove any odours. Would you like me to use one of these?
Yes, that would be helpful.
Okay, I can do that.
You haven't got one that could tell if it's infected, have you?
Not yet, but there is one that's being designed to do just that. A dressing that would change colour in response to increasing bacteria in the wound. Hopefully that won't be too far away.
A dressing that would change colour if it's infected, that would really help you stop worrying.
Yes, it would be helpful, as if you notice changes in colour, you would know to contact the hot foot line straight away. But until then, we need to look out for the usual signs of infection.